Thanks for your attention.
Great. Let's thank Timo for a great talk. And let's go straight to questions. Our first question is from Tom.
Thank you, Timo. That's inspiring stuff. Would you expect in a passive system where you're expecting to get this half, that if you've got some kind of structured environment where you expect whatever particle you're tracking to be undergoing subdiffusion, that you kind of, if you measure on timescales that are too short for it to escape its local environment, you get a half. And then if you measure on longer timescales in which it can start to move around the cell, you move away from a half. But then if you go for really long and you actually look at the whole cell, when you actually see the confinement of the whole cell, then in theory you'd expect to get the half back. Is that consistent?
Yes, this is totally consistent. I mean, honestly, what you just point out is why when we started looking at the cells, we didn't even expect this to work, right? Because the cell is a length scale that for these particles to see that would require potentially hours, right?
Yeah.
So the reason why it seems to work for us is that really we have a local confinement that is strong enough for it to hold. And if there would be, just as you described, right, if we would be on a situation where suddenly it hops out and it becomes kind of like freely diffusing, it would also go down. So it was not at all trivial that it works. And
you must be that just isn't that important on the time scale that you're looking at.
Yes, exactly. This is the thing. So we look at the moment typically at timescales here of about a second, right? We record the trajectories typically for like 100 seconds to get good statistics. But the time scale that we look at here is just a second. And if you look for very long times, then also this data becomes more dirty back there, right? In the cells, because the cells are not necessarily your friend, to be honest.
Okay, we've got a queue of questions, so let's go straight to Rui Cheng.
Hi, very cool talk. So my question is, in general, is the larger deviation of MBR from 1/2 implies larger entropy production rate?
Yes, I think so, but you have to be very careful because the entropy production rate, of course, requires, the way we derive it at the moment, requires the antisymmetry. So we're looking at this, I cannot guarantee you that it does it. I would be very careful. If we do this in these more controlled systems where we have a potential and we move this around, then it's totally fine. Inside the cells, I'm always a little bit more cautious.
Yes, I noticed that in your analytical example, this is the case.
Yeah, I think so.
Thank you very much.
Okay, let's go one quick question from Stephen, and maybe Heiner can set up his slides and Timo can unshare as we take the question.
Right.
Yeah, my question is about sort of the length scales involved. Do you look at all at like the inhomogeneity in the spatial structure, and are you able to resolve like spatial correlations between within these systems?
Yeah, this is an important question, of course. So what we do at the moment, we neglect this, right? Which means we have to pay the price of doing like huge statistics. So we have like hundreds of experiments individually, and the error bars are annoying. And for a physics master student, it's very hard to motivate them to come to it. But if you kind of like do the statistics right, you recover quite nice curves. This is what we try to do now in the future, of course, looking at correlation function, especially looking at spatial directions. There's lots of inhomogeneities in there, but you have to have a very carefully designed experiment to actually look at this because every cell is different, right? But yes, definitely right. And there's a lots of stuff to be done.
Yeah, thank you.
Thank you.
Cool. Let's thank Timo again for a great talk. And we will move on to our last but not least speaker of the day, Heiner Linke.
Yeah, thank you so much for the opportunity to speak here and also for all the great talks before. Great to listen. So this talk is about a project that's quite exciting for me and my colleagues because we worked for many years to realize this and are now very happy that we can report that we have made an artificial motor protein, which has been an aim for us for a very long time. That, yeah, as I show in the title, can walk along a track. So I will give a little bit of background first and then try to explain this beast. but also in the end, where we see some opportunities to study stochastic thermodynamics with the system, because that's one of the advantages of having an artificial system, that you know exactly what you put in there. So you all are familiar with motor proteins. This is a clip or a still from this famous animation of what's going on inside the cell, showing or illustrating, I should say, a kinesin motor transporting a vesicle along a microtubule. And this is also the inspiration for this project. There's of course many activities have been done to study motors and also to make artificial motors. We are specifically interested in protein motors, so in the original, so to say, because on the one hand, of course, there's all these important functions in the cell, which I'm sure you're aware of, that essentially every process in the cell in one way or another is powered by molecular machines with a very high degree of complexity and variation in what they can do and how they work and what they look like. They are also very cool from a physics engineering perspective because they are surprisingly efficient. So if you look at the conversion of chemical energy into work, they are at least comparable to man-made combustion engines, and one can argue that they beat them in many cases. And probably not in spite of the fact that they live in a world of thermal motion, but because of, and of course you guys are all aware of this. And so I will also try to highlight in the end, again, that there's also, so these are of course, in many ways, the inspiration for stochastic thermodynamics originally. They are clearly governed by stochastic thermodynamics, and maybe I can show also a system to study them. And there I would also welcome any comment or contact after the talk maybe about ideas what to do there. So we started this project many years ago, and the inspiration is basically these stepping motors that can walk along a track. This is a simulation from a paper we wrote a number of years ago about myosin 5. And what we try to do is to mimic a motor that can walk along a directional track, like this myosin 5 that walks along actin, which requires us to have some kind of coordinated binding and unbinding, coordinated in the sense that one foot needs to let go while the other one is still on there to achieve processivity. So processivity is the term for staying on the track for at least a significant number of steps. Thermal motion, of course, is always present. And generally this means for a biological motor that you need to go through a cycle of ligand binding states. So in a ligand is a small molecule that often controls binding and unbinding affinity of another molecule. And in biology, that's of course ATP and its products in the hydrolysis. And that enzymatic cycle in a biological motor sets also the timing. As you will see, we have not achieved all of these, or at least some of them we need to do artificially. So far, so the concept that we've been pursuing for quite a number of years, as you can see there, is to say, let's do part of this from the outside. So this is not an enzymatic motor. It's not an autonomous motor. We need to control it from the outside. And the basic concept is to use use proteins that by biology are designed to specifically bind to a specific sequence of DNA. So those are used in gene regulation. And there are proteins that are regulated themselves by smaller ligands in solution. So basically the red ligand here, which is called A, or the red protein that is called A, will bind to its specific sequence of DNA in the presence of a ligand, little a. And then if we have 3 of these, they need to be different. They need to have different binding sequences on DNA, and they need to have different ligands. So if we then, for example, offer the ligands A and B, then the proteins A and B, the red and blue one, would then bind to their specific sequence. And then by cycling through the 3 ligands in an appropriate way, the motor would start walking, entirely driven by diffusion. So the stepping itself is then done by the motor diffusively finding its next binding sites without any power stroke or anything like that. And in a model, you can realize this very, very quickly. You just tell the thing to bind when you tell it to bind, and then you would expect something like this, where you diffuse around looking for your new binding site, and then waiting for the next cue that you can unbind and so on. And of course, you would get, if you set things up nicely in a model, of course, you can get pretty much perfect stepping motion. In doing this in practice is a little bit harder. So this is the process that I show you now, just from the protein, on the protein design part, took more than 10 years to even get to the point where we're able to do single molecule experiments, but now we have the whole package. So I abbreviate this very much, but here you see a pi mole realization of what our beast would look like. So it consists of 3 proteins. They are called here which we call the feet, METJ, TRPR, DTXR. They are proteins that exist in nature that bind to DNA on specific sequences, which we have spaced here by about 50 nanometer in such a way that the protein would bind from the same side onto the DNA. The DNA has a helical structure, so you need to make sure that you don't come from different directions. Each of these proteins has their own ligand that you don't need necessarily to remember, but they are SAM, a small molecule, tryptophan, and cobalt ions simply, for DTXR. Yeah, and then we need legs to get the spacing. So these are alpha coils that we have also stolen from another biological system. And the hub in the middle that joins it all together, which gave actually a lot of headache for many years until we found the right system for this. Yeah, so I will now sort of break this down in a 4-step approach to show how we got there. So we need to build it. We need to make sure it binds when it's supposed to bind and unbinds when it's supposed to unbind. We need to control it, meaning we need to supply those ligands in the right time sequence and reliably. And then we need to observe it. We need to actually see whether it steps or not. So I walk you through this in steps. We build it, since I assume there's not many molecular biologists or protein designers in the audience. I will keep this very short, but this took very long. This is many years what I show here. The system that worked in the end is a trick where the hub consists of 2 parts. That is a molecular biology system, spy tag, spy catcher, 2 parts of a protein that when they meet actually form a covalent bond, so a very strong bond that you can then use. So we express this protein in 2 parts. So E. coli cells make this by instruction. So there's a gene sequence you give it the sequence that defines this protein structure. So we call that tumbleweed 1. And then the other one, tumbleweed 2, which contains a foot, a leg, the spy tag hub part, another leg, and another foot. And then once both of them are expressed, you can bring them together in solution and they will join together. And then there's a number of things you can do to verify that this probably happened. You can look at mass, you can look at size and so on. And we can verify that both the parts and the combination look like they do what they're supposed to do. The first binding experiment that we did was also on when we still thought we would use long DNA tracks to actually observe motion. So this is a long DNA that comes from virus, modified to contain, not exactly in the middle, but at the point that you can see here as bright spots, 8 repeats of these 3 binding sequences that we would need to make tumbleweed walk. So it's a total of 24 sequences spaced in the appropriate manner. And we loaded tumbleweed on this in the presence of one of these ligands so that one of the feet would bind, and can then see in AFM that indeed we get 8 blobs at the correct location with the correct spacing on this DNA here, which was the first verification we had that we can actually see this. Then there's a lot of experiments, in particular in recent years, performed by our collaborators in Sydney, where tumbleweed was built. a method using called SPR, surface plasmon resonance, which is basically we use a monolayer of DNA on a surface, and then very sensitively can determine mass, and in this way verify whether something binds to the DNA. And so there was a lot of experiments done, but the key thing here is to control and to verify that you have what we call orthogonal binding, meaning tumbleweed has these 3 feet. And for example, the MET-J foot, which is supposed to bind in the presence of SAM, should bind when you offer that ligand, but should not bind when you offer any of the other ligands, and vice versa. So you want this, when tumbleweed has no ligands, nothing should bind. And then you want to see this diagonal here, that each foot bind when it's supposed to, but not, or at least very, very little when one of these other ligands is offered. So this was verified in the end. This took, of course, some optimization, but also we will be able to then optimize all our concentrations, and also to measure our unbinding and binding rates, which is then important to set up the control of the stepping appropriately. So these experiments were very important. Yeah, and then we need the control. So basically what we want is to persuade tumbleweed to bind with 2 feet, for example, in the presence of the 2 ligands, tryptophan and SAM. And then we want to be able to switch to a solution with 2 other ligands, SAM and CO, so that one of the foot would let go and another one would bind. And then you need a third one, CO-trip, for the final pair of 2 feet to bind. And then you need to go back to trip SAM without going through the solution SAM-CO. So you want to keep going, which required us to come up with this slightly unusual microfluidic device, where we, for each of the 3 buffers that we need, we have 1 pair of channels, an entrance and an exit, which allows us, by pressure difference here, in arbitrary time order, to control what goes down this main channel where the observation is set up. So tumbleweed in the end would be at the bottom of the channel here along this long bit at the end, and so that we can look at it with a microscope. And here you can, with dyed buffers, so we use no dye, a medium concentration and a high concentration of dye. So you can see how this switching works. You can see that you indeed can do pretty quick transitions between buffers in this main channel. So we can show that we, even though we are diffusion limited at the boundary where tumbleweed actually sits, we can switch with at least 0.2 second time resolution, which we won't need in these experiments, but this is where we can go. Yeah, and then the observation. So the experiment that I can show you is based on FRET. So FRET is a technique that allows you to look at, by fluorescence, whether for example, 2 parts of a protein, or in this case, a protein and a DNA point, are very close to one another, because the fluorescence of a donor gets transferred to an acceptor. So I tried to explain how this works. So we use a surface with short pieces of DNA that contain 4 binding sites for the motor. Trip is there at the beginning, at the end, and then you have the 2 others in the middle. So you can take 2 full steps. You can go from bound trip to DTXR, to DTXR and MET-J, to MET-J and trip R. And then we can, in principle, do this in reverse. You can walk up and down this track if you set it up nicely. To verify motion, we put a fluorophore, the green one, on one of the feet, on the trip R foot of the motor. And then you expect the following. If the foot is bound to the lower end of trip R, you expect FRET, so an energy transfer from the green donor to the yellow acceptor. So you would see a yellow signal. If the tryptophan foot lets go and is bound in the middle, you would expect the green signal, so the donor signal unperturbed. And then when we bind to the other end of the DNA, you would expect FRET again and a red signal. So in principle, we have, for each of the 3 possible binding situations, we have 3 different signals. Unfortunately, our microscope only allows us to see 2 colors simultaneously with high resolution. So we did 2 experiments. One where we distinguish whether it is at either one of the two ends. Both of them would give one color, red. And the third one would be if the motor is in the middle, it's green. So if one steps up and down, you would expect red, green, red, green, red, green, red. But you cannot see whether it actually is at the top or at the bottom. To distinguish that, we did a second experiment where we distinguished the top and the bottom, but have a dark state when the motor is in the middle. So I will show you both of these experiments. So this is a single, so this is basically on the left side, you can sort of see, I guess it's not very bright maybe, what you actually see in a microscope. You look at a field of view, you see bright spots where molecules sit, and we have several hundred of them in a field of view. And then you can isolate spots where you actually see DNA and tumbleweed in the same spot. There's still hundreds of them. It's a fraction of them, but still hundreds. And then you can make time traces of one of the luminescence from one of the spots as we change the buffer. So we change buffer every 7 seconds. And what you then can see, if you look at one of the spots, you can see how the red signal goes up and then down and then up and then down, and the green signal alternatingly. And this is exactly what we expect, because we want to see the motor go from the bottom to the middle to the top to the middle to the bottom and so on. And we just keep stepping until we don't see anything anymore. When they go dark, one of two things has happened. Either tumbleweed has fallen off or it has been bleached. The fluorescence can bleach. And we set things up that we would use maximum time of our fluorescence so we can really not distinguish them. But we can sort our tracks here. So each of these lines here is one fluorescent spot, and you see very nicely that there's an overall pattern of red and green. When we resort them by how long do we see each of these spots, then you get also a very nice decay pattern where you can see some motors fall off or bleach very quickly, some stay longer, and we see up to 11 steps on a single motor. And as I said, what we haven't shown with this yet is that we actually reached the top and the bottom, but this we can see with this experiment where we now distinguish the top and the bottom markers, and in the middle, the motor simply goes dark. And now we see indeed the two colors alternatively with black stripes, but we also see more complex behavior. In one of the bright tracks, when the motor is supposed to be, for example, at the bottom, we still sometimes see it switch to the color of the top. And what we believe is going on is that our DNA is flexible, and in this state where one foot is supposed to be bound to the bottom, it's dynamic binding. It will occasionally come off, and then rather than rebind where it's supposed to, it seems to be able to reach the top in this sort of bend situation. We can then do statistics on this and actually work out how much time it spends in each of the two states. So this is an imperfection, which is also not uncommon for this type of system in biology, of course. But that has to do with the flexibility of our track here. Then because we think we have very convincing evidence that we are stepping, but we are only walking up and down this very tiny track. So what we have done is we set up a model, or this is done by Ralf Eichhorn at Nordita, a model that simply says, what if we had a motor that behaves exactly as we observe it, because we can measure all the rates, we know all the binding and unbinding rates and everything. What if the track was actually extended? How well would it perform then? And we can do this to either include this overstepping that we observe or not, and can then sort of predict what kind of performance would this motor have on an extended track that doesn't end. And as you can see, with the overstepping, we see there's still forward motion on average, but there's very large variability. This is what you would expect, because it sometimes basically can walk backwards also. Whereas if we could go to a stiff track, or we assume we go to a stiff track where this overstepping wouldn't happen anymore, then even with the rates we have now, with the timing we have now, we would expect a pretty deterministic motion. But this also highlights something for us, namely that there is a degree of unreliability that can happen that is simply, by unreliability, I mean this sort of high variation that you observe here. So that can either be due to experimental imperfections that simply happen now and then, like in this overstepping case, On the other hand, we also expect, simply because this is a stochastic system, there should be variation. So we got interested in the question whether we can distinguish the two and we can understand how much variation is fundamentally expected and how much is only because of some other factors, which I get back to in a moment. I forgot the slide in the middle here. We looked also at the speed and the processivity that we observe at the moment, which nicely puts this first protein motor between what has been achieved with DNA motors so far. They tend to be a bit slower, but still well away from the biological motors. The red dot here is what we have observed so far. We think with faster switching, we should without problems be able to go both faster and with higher processivity, simply because we wouldn't fall off so quickly. So we would be able to run more steps, but we haven't shown this one yet. Yeah, but back to the variability, we got interested, is this where would be the limit in terms of thermodynamics, what we can expect in terms of reliability? And started from these very nice thermodynamic uncertainty relations, which in this context would mean that the entropy production can be related to simply the energy consumption, assuming that the chemical energy that you put in, if you don't do work, if you just walk along the track, will ultimately end up as heat. And that is interesting to us because it allows us to put the chemical energy input, so in this case, the chemical potential difference that drives our motor when we supply ligand at high concentration, remove it at low concentration, that we can relate our energy input directly to the expected diffusion in this case. And we say, if we can take, so to understand, to define what is it we're actually interested in, we want to say, for our motor to have a certain performance, we want to be able to say, take n steps of a size d. So our d is about 50 nanometers. For tumbleweed, we see about 10 steps. And then we see in our experiment an uncertainty of about plus minus 1 steps or so. So this is about what we see at the moment. And an interesting question for us is, of course, is this as good as it gets, or do we simply have experimental problems? Yeah, and so Elena Rufail, who I think is also on this call, worked on this, and one can relatively easily then simply rewrite the TUR to express the energy input, our chemical potential input, our delta mu, to what we picked as a parameter, a randomness parameter that very early on in the research on molecular motors was defined by Steve Block, which basically combines the step size, the speed, and this diffusivity. And one can also translate it into this K and N that are defined up here. So for us, we would want to see what chemical energy input do we need to see this performance of a K of 1 or 2 divided by N. And this gives them pretty, if you write this down, you can plot this. This gives them, for an experimentalist, a pretty interesting plot, because you can basically read out, if I have a chemical reaction that gives me a certain free energy, or in this case expressed as a chemical potential difference of say 10 kT, then you can read out that you cannot hope to do better than this randomness parameter. So you will only achieve, so a randomness parameter below 0.2 in this case is simply not possible, while higher is possible depending on the performance on your system. And as a red dot, we marked roughly where our tumbleweed is at the moment. So this basically tells us we need an energy input of at least 10 kT to be able to see what we are seeing. And what is our energy input? I'm running out of time, so I keep this very brief, but basically one can pretty simply estimate it, because what tumbleweed does is it takes a ligand at high concentration and spits one out at low concentration. And then we change the buffer, and it again takes a ligand at high concentration, spits one out at low concentration. So if one knows all of these concentrations, one can work out what delta mu we are actually offering. In our case, we are actually trying to have the low concentration as low as possible, but we can read out that for a ratio of high to low of about 10 to the 4, we would be able to get the performance that we see. So this gives us sort of a bound of what thermodynamics does. And one of the things we would like to try now, there's many things to do, but one of them is to actually change our concentration so that we can control our energy input and see how performance changes. There's many other factors in there as well. In reality, our concentration changes are not instantaneous. There's a time dependence in there, so there's many things to factor in there. Yeah. So we want to do that. We want to see how fast we can go. We are working hard to make stiff tracks now using DNA origami to get rid of this overstepping. And our real aim, where we are working on in our whole big team, is to get to autonomous motors. But again, these considerations based on the TOR are really useful for us in choosing candidate systems for enzymes, because we can see how much we can now actually calculate how much delta mu, how much delta G we actually need to get a certain performance. So this is very useful there as well. And finally, I want to thank all the people who have worked on this over the many years. So this all started with an HFSP project in 2008 and now was completed with an ERC project in recent years with long, hard waiting times in between and work from many, many people. Yeah, thank you very much.